Okay, wonderful. Then, without further ado, I think I will start. So first of all, thank you very much, John, for the introduction. And also thank you very much for inviting me to give today's seminar here at the Astbury Centre. And it's actually the very first time I have been invited to give a talk about my research. So I'm very happy and excited about that, and also feel a little bit honest about it, to be honest. So thanks very much again. Yes, so in today's talk, I will present a project which I did during my PhD at the Rendell Centre at King's College London. And the talk will be about phospholamban, a small heart muscle protein, and in particular about its pentameric form and how it processes the information in this pathway. So before I go on to any biochemical details about phospholamban, let me briefly introduce you to the physiological context in which phospholamban plays a role, which is the regulation of heart function. So the function of our heart to pump blood relies on the ability of its heart's muscle cell to contract and to shorten. And on a molecular level, the translation of electrical activity into mechanical force is caused by a process called cardiac excitation-contraction coupling. And it briefly goes somewhat like this. So, whenever the heart receives electrical activity in form of an action potential, which can propagate along the cell membrane and deep into the tubules, this electrical activity can then cause the opening of calcium, of voltage-gated calcium channels, which leads to an influx of calcium ions from the extracellular space. This calcium then triggers the release of further calcium ions from the cell's internal storage compartment for calcium, the sarcoplasmic reticulum. And together, this leads to a wave of calcium within the cardiomyocyte that then can activate the sarcomere, the myofilaments in the sarcomere, to enable it to perform cross-bridge cycling and to generate force. And therefore, the heart muscle cells and the whole heart can contract and eject the blood out of the chambers in order to perform its function. For the next heartbeat to occur, obviously, the cell and the heart also needs to be able to relax so that the chambers can refill with blood. And that means that all the calcium needs to be removed from the cytosol of the cell again. And while some of it is pumped out into the extracellular space, the majority is going exactly where it came from, namely the sarcoplasmic reticulum. And that's caused by the activity of a calcium ATPase called SERCA. And once enough calcium is removed, then the heart relaxes, the chambers refill with blood, and the whole cycle can start all over again. Our heart also needs to be able to adapt to different levels of demand in different physiological situations. So for instance, when you're in a stressful situation, your heart needs to pump faster, it needs to pump stronger, but it also needs to be able to relax faster. And one of the most important signaling pathways to regulate heart function on short timescales is called the beta-adrenergic signaling cascade. So when the cell receives stress signals in form of adrenaline or noradrenaline, these molecules can then bind to the cell surface receptors of the cell, which then triggers the activation of a cascade that ultimately leads to the activation of a kinase, protein kinase A, or short pKa. And while pKa can phosphorylate lots of different important targets in heart muscle cells, one particularly important one in this context is called phospholamban. Phospholamban is a small protein that resides in the membrane of the sarcoplasmic reticulum, where in its unphosphorylated state, it binds and inhibits the SERCA pump. When it becomes phosphorylated by pKa, this inhibition is released, and therefore the activity of the SERCA pump is increased. And that means at the end of a contraction cycle, the calcium is removed at faster rates, and therefore the heart can relax faster. It also means that at the end of such cycles, the filling of the sarcoplasmic reticulum is higher with calcium. So the next, or the amplitude of the next calcium transient will be bigger, and therefore these contractions will be also stronger. So what I found particularly interesting about phospholamban is that it forms these, frankly, quite beautiful pentameric structures here. And despite over decades of research, no one really knows what these pentamers are actually doing. Obviously, there have been some ideas in the literature, and one of the first concepts was that maybe it's something like an ion channel. And if you look at the NMR structure here, that intuitively makes some sense, because if you look at this alpha helical bundle, the transmembrane region of the pentamers, you can see something which looks like a pore. So that's something which at least structurally looks like it makes sense. And some groups have also reported that there is some ion channel activity. However, the experimental results regarding the selectivity for different ions are somewhat contradictory. Other groups have found no channel activity at all. And the whole hypothesis has also been, or this theory has been challenged based on thermodynamic arguments. So possibly, or yeah, probably it's not the most convincing theory, at least at the moment. And because it's been also demonstrated that the monomers are in principle completely sufficient to bind and to inhibit the Serker pump, and that they can also be regulated by phosphorylation and dephosphorylation, it has been proposed, or that the most dominant paradigm in the literature is actually at the moment that these pentamers are just some inactive storage form or buffering form that somehow keeps the concentration of monomers in a physiological range, but that there's no active role for pentamers in heart function. However, given the previous context of beta-adrenergic signaling, I, for this project, asked a very simple question, which is, how do pentamers influence the phosphorylation in the context of beta-adrenergic signaling? And I'm not the first to ask this question, but one of the first who was actually John. So he and his group in the early 90s studied phospholamban quite extensively, and he believed that the pentamers may actually offer an effective substrate structure for both kinases and phosphatases to influence the regulation of phospholamban phosphorylation. And the reason for why he believed that is that he and his group characterized the pentamer of phospholamban as a substrate for various enzymes. So for instance, he characterized, or he followed the pattern of differently phosphorylated pentamers on the gel. So when you phosphorylate pentamers, you can see different bands for different phosphostates. And he looked at the pattern of these experiments. And what he found, for instance, for pKa is that the distribution of the phosphostates of the pentamers closely resembles a binomial distribution. And that's something which you expect for a random phosphorylation mechanism in which the phosphorylation state of the different subunits within a pentamer don't influence or doesn't influence the phosphorylation state of the other subunits within that pentamer. And when you compare the distribution to a theoretical distribution, you see a very close alignment, which suggests that indeed pKa phosphorylates pentamers randomly. In contrast, he found that the dephosphorylation after complete phosphorylation has a quite different pattern. So at 50% dephosphorylation, he found that this pattern looks strongly U-shaped. And that indicates that for dephosphorylation, the phosphorylation state of individual subunits within the pentamer does influence the state of other subunits. And in particular, it suggests that the more phosphate groups you remove from a pentamer, the faster the rate of the removal for the next phosphate group will be. So what you get is that over time, the more phosphate groups you remove, the faster this reaction becomes. So it's strongly positively cooperative. There also have been other interesting phenomena described in the literature, which somehow relate to both the phosphorylation and the pentamerization of phospholamban, one of which is called the dynamic equilibrium. So phosphorylation seems to increase the pentamerization of phospholamban. And that's something which I could also confirm myself in the lab when I solubilized the phospholamban peptides in SDS-based buffers and phosphorylated them with pKa. I found that the ratio of pentamers to monomers actually increases. So yeah, that's another interesting phenomena. And what the Schmidt lab in Germany has found, moreover, is that if you express phospholamban in egg cells and stimulate them with an agonist which activates pKa and follow the phosphorylation response, then you can see that the phosphorylation of pentamers is stimulated at rather low doses of the agonist and saturates quickly. But in contrast, the monomer phosphorylation is increased only at higher doses of the agonist and also saturates later. And interestingly, if you harness an artificial mutation which prevents oligomerization, you can see that the phosphorylation is triggered at lower doses and also saturates earlier. So that suggests that pentamers somehow attenuate the phosphorylation of monomers in these cells. Now, for my study, I decided to follow a somewhat different approach to what others have done so far with phospholamban, which is to develop a mathematical model of the phospholamban signaling network in order to make sense of all these different phenomena. And in this model, I included the circa inhibitory species, or the monomers, and its phosphorylated state. I also included the pentamers and how they can be phosphorylated in their different states, and of course all the relevant enzymes in this context, which is pKa, but also the protein phosphatases PP1, PP2A, and also an feed-forward loop, which features in the inhibitory protein inhibitor 1. I didn't include anything further upstream of this network or further downstream because I only wanted to focus on the phosphorylation state, and then described everything using ordinary differential equations and yeah, used parameters from the literature in order to set the model up. And once I had this model, I obviously wanted to use this to perform some simulations. And one of the first simulations I did was a very simple one. So I just took phospholamban, added some kinase to it, and yeah, without any phosphatases present, and just followed the time course of the phosphorylation. And what you can see here is in the blue line that the pentamers are phosphorylated rather quickly. And in comparison, the rate of monomer phosphorylation, shown in red here, is happening at a much slower pace. Interestingly, if I repeat the same simulation without any pentamers, you can see that the rate of monomer phosphorylation is much faster. And this is because in the model, the pentamers and the monomers compete for each other, with each other for phosphorylation by pKa. So you have a substrate competition situation. Now, because there's this nice artificial mutation which you can use to prevent oligomerization, you can also test this model prediction quite easily, and that's exactly what I did in the lab. So I first looked at the phosphorylation of monomers in the presence of pentamers, shown again here in red, and then used another experiment where I had the same concentration of monomers without any pentamers, and then also phosphorylated them using pKa, and I found that the rate is much higher. So the pattern you see in the experiment qualitatively nicely matches the simulation and therefore confirms this model prediction. So in addition to these time course experiments and simulations, I also wanted to know what happens at steady state as a function of pKa. And the reason for that is that I found in a previous study, in a theoretical study of mine, that when you have oligomeric proteins which can be phosphorylated, then all sorts of really interesting phenomena can happen. And in particular, I found that if you have an oligomer which can be phosphorylated, then essentially you have a pseudo multi-site phosphorylation system. And we know from other studies in systems biology that multi-site phosphorylation systems, for instance, can generate bistability. So the graph here at the right, you can see if there's a modifying enzyme such as kinase, and if you increase the activity, then what can happen is that you get a switch-like response from a low phosphorylation state to a high phosphorylation state. And if you go the other way around, then you can also get a switch, but as a different threshold. So within this region here, this bistable region, the situation can be compared to like a little ball sitting between two valleys, and the ball can end up in either of these steady states, depending on where the ball falls down into from where it falls down into one of these valleys. And because John, with the positive cooperativity for dephosphorylation, has found exactly these kind of kinetic requirements for such phenomena to occur, I implemented this positive cooperativity in my model and for phospholamban, and wanted to know what happens in this case. And what you can see in these graphs is that the phospholamban model also predicts both the pentamers and, because of the competition, also the monomers to be phosphorylated in a switch-like and bistable fashion at steady state as a function of pKa. What I also found is that that the parameters for cooperativity and also for the dynamic equilibrium are actually quite important to shape these dose response curves. And in particular, they strongly determine the bistable range, so how big the area is in which you can get bistability, and also basically the size of the jump from the low to the high phosphorylation state. So both these phenomena seem to be really important to shape these interesting dose responses. Now, when you have such a model prediction, you also want to make sure that this is not just some numerical prediction. You also want to know whether that's actually something which occurs in real biological living systems. So I first focused on the switch-like behavior of the system and looked at different dose responses which have been already published in the literature. And I wanted to measure this degree of switch likeliness, also in systems, which is in systems biology also often called ultrasensitivity. And the way to do that is often to fit these dose responses to Hill equations and then look at the Hill coefficient. And if it's greater than 1, then you have some degree of ultrasensitivity at least. And what I found is that indeed in several of the published dose responses, I could find Hill coefficients of about 2. And in particular, what was quite interesting was that this study here from the Schmidt lab found that this ultrasensitivity also seems to depend on the presence of pentamers. So when they use this mutation to get rid of the pentamers, then the switch-like response was basically abolished, and you get a Hill coefficient of about 1, so without any switch-like behavior. Because in these experiments, unfortunately, the pentamers started at a very high basal phosphorylation level. I asked Florian, which will give the second talk today, also to be so kind and to repeat these experiments for me. Yeah, he did. And what he found is that, again, the Hill coefficient was roughly about 2, a bit higher for the monomers, a bit lower than for the pentamers. But in generally, he could again confirm this high Hill coefficient. And interestingly, this seems to be not only restricted to the phosphorylation, but also something which can be found at the level of physiological parameters as well. And in particular, he generated a transgenic mouse model with only monomeric phospholamban, no pentamers, and compared that to the wild type situation. And when he looked at the relaxation of the heart as a function of beta-adrenergic stimulation, he also found that the slope basically of the curve for the mice without pentamers was much shallower than for the wild type mice. And in addition, he found that also the range of regulation is different in the presence of pentamers. So yeah, he will tell you a little bit more about these experiments himself in the next talk. But generally, that also seems to hint into the same direction. In addition to the switch-like response, I also wanted to know whether there's actually bistability happening in phospholamban phosphorylation. And the way to do that is to test whether phosphorylation exhibits hysteresis. so that the steady state of the system of the network which is approached depends on basically the initial state. And how I did this was that I took neonatal rat cardiomyocytes and stimulated them with isoprotenolol and beta-adrenergic agonist. And I split the cells into two different groups. So one group was not pre-stimulated with any agonists, and the other group was shortly pre-stimulated with beta-adrenergic stimulation. And then the dose of the agonist was adjusted to the final dose, and the cells were given enough time to reach steady state phosphorylation. And then I compared basically the phosphorylation levels between these two groups at different end concentrations of the agonist. And what you can already see from the plots here is that at some concentration, the cells which have been pre-stimulated seem to retain a higher level of phosphorylation. And when I quantified the data, I saw a very nice hysteresis loop. So the arrows here basically indicate the direction in which the cells were stimulated. So when you go up, you follow these arrows here basically. And when you go down from the highest phosphorylation state, you go the other way around. And then you can see at a concentration which is slightly below the EC50 of the dose response, you can retain a higher steady state phosphorylation for pentamers, which is basically a direct confirmation of this bistability, which was predicted of the model. And since the model also predicted monomers to be phosphorylated in a bistable fashion, I also wanted to test to test this, of course. But unfortunately, I wasn't able to separate under my conditions the monomers from other small oligomeric forms. So what I did instead is I lumped all these different forms together as low molecular weight forms and quantified them, but I couldn't see any significant statistically significant difference under these conditions. So I think in the future I need to optimize this experiment a little bit in order to reach a conclusion. But at least conceptually, and based on the model prediction, I would also expect to see at least some hysteresis for monomers as well. Now, so far I've been waffling on about quite puzzling phosphorylation-dependent phenomena for quite some time. So the really interesting question now would be, of course, what is it all good for? And after pondering this question for quite some time myself, I came across this paper here from the Kranias lab, in which they describe or discovered a mutation in the phospholamban gene in patients which had a lethal form of cardiomyopathy. And in particular, these patients had a very high prevalence of cardiac arrhythmias. And by characterizing a mouse model of this mutation, they also found that it impairs the phosphorylation, but it also destabilizes the pentamers. So I was wondering, what if pentamers may be playing a role in preventing cardiac arrhythmias? So yeah, the next question would be, how would they actually do that? What's the mechanism behind this? And that's when I remembered some high school physics lessons about electronic circuits. And in particular, I remembered that these delayed delayed time courses, which I saw for the monomer phosphorylation in my simulations, is also something that we know happens in electronic resistor capacitor circuits. And these circuits can be also used as signaling filters. And in particular, they can act as a low-pass filter. So that means if you subject these to signals of different frequencies, then basically high frequency signals will be effectively filtered out, and all the lower frequency signals will be passed on. And I was wondering whether phospholamban pentamer or pentamers or the phospholamban network in general also has such noise filtering features. So I performed some further simulations in which I subjected the model to a fluctuating kinase pKa activity consisting of a slow component shown in red here and some fast random noise shown in black. And I then followed the phosphorylation of monomers as an output of the model And what you can see here on the right-hand side is that the phosphorylation output closely resembles the slow component, but that all the fast random noise and kinase activity is effectively filtered out. Now, in order to quantify the contributions and the relevance of pentamers in this context, I performed what is called a frequency response analysis to determine the bandwidth of the system. And what I found was that in absence of pentamers, the bandwidth is much higher than in a model that still has these pentamers. And that means that much more of these fast random noise signals are actually passed through and not filtered out. So taken together, these simulations confirm that the signaling network of phospholamban does have low-pass filtering function, and that these pentamers also contribute to it. Now, since this only gives a plausible explanation for the response delays, we also need to ask what the function of bistability would be. And I thought that maybe bistability may be actually acting in a very similar fashion, but maybe on different time scales. And the idea for that would be if you compare the dose response of a bistable model shown in the red curve here, and with a model that doesn't have any bistability shown in purple here. And if you then have fluctuating kinase activities as indicated here by the arrows, then without bistability, it could be possible that you constantly fluctuate between a low phosphorylation state and a high phosphorylation state. Because the dose response closely follows the fluctuations. However, if you introduce bistability, some of these fluctuations may be filtered out, and only if you pass these two distinct thresholds, you actually get a switch from a high to a low phosphorylation state, or the other way around. And in order to make sure that the fluctuations which I'm looking here at are not already filtered out by the low-pass filter, I subjected the model to basically very slow fluctuations to make sure all these fluctuations can cause their effect. And when I then compared the bistable model shown here in the red curve with the non-bistable model, I found that the phosphorylation state in the presence of bistability is generally much clearer defined and switches far less frequently. Whereas when you don't have bistability, you really can constantly fluctuate between a low and a high phosphorylation state. And if you quantify the switching events, you can see that these events are actually, the switching happens about 5 times more frequently. So yeah, this also confirms that bistability could be a way to filter out fluctuations or noise in this signaling network. So my current hypothesis is actually that this low-pass filtering and the bistability complement each other in order to promote a clearly defined phosphorylation state of phospholamban in the presence of fluctuating kinase activity. And if you remember that phospholamban is really an important regulator of SERCA and calcium cycling protein, and that calcium is well known to be an important factor in the pathogenesis of cardiac arrhythmias, I think that this noise filtering may be actually a way to ensure cardiac coordinated calcium cycling in heart muscle cells, and therefore maybe prevent arrhythmias. And there's also some more evidence to this idea, because it's known that mutations in other model components, which also contribute to this low-pass filtering behavior or noise filtering behavior, actually do cause cardiac arrhythmias. But at least for the bistability, however, it's also possible that there may be other functions, physiological functions to it. So one idea could be that if you have, if you want to maintain the beta adrenergic response for longer time periods, then this may be actually a mechanism to retain high inotropy or lusitropy, so meaning stronger contractions and faster relaxations, despite maybe waning inputs or decreased inputs from the upstream network, because, for instance, it's also known that there's some adaptation, at least on the receptor level. So you could maintain maybe this response for longer periods of time. And with that, I'm already at the end of my talk. So in summary, I've shown you that pentamers shape both the dynamics and the steady state of monomer phosphorylation, and that they may act as molecular noise filters to ensure consistent circuit activity. And in the future, I think it may be really interesting to investigate whether beta-adrenergic, noisy beta-adrenergic signaling may actually cause or trigger cardiac arrhythmias. And I would also like to know whether hysteresis following beta-adrenergic stimulation actually does occur on the level of monomers as well, and whether that's in particular something which you can see in the calcium dynamics. And if that's the case, then stabilizing pentamers may actually be an interesting novel therapeutic strategies, maybe to prevent cardiac arrhythmias,